Bismillahirrahmanirrahim. So, after a very enthusiastic first session in the morning, we have moved to the second session for today. So, it's an honor for me to moderate this session. My name is Dr. Qadir Sakarwala. I'm working in SIUT. Uh, so, again, it's an honor for me to introduce the first chair. Once upon a time, but she is here, Dr. Ghazala Jafri. She is a retired professor of pathology from Services Institute of Medical Sciences, Lahore. She has made significant contributions to the field of medicine and immunology. Her academic journey includes earning her MBBS degree from King Edward Medical College, Lahore, and a PhD in immunology from Victoria University of Manchester, UK. She has more than 40 research publications. Dr. Garala expertise extends to editorial responsibilities as respected member of many editorial boards. Additionally, she has held esteemed position of Vice President in the first Executive Council of Pakistan Society of Immunology. She is one of the founding members of Pakistan Society of Immunology. This showcases her dedication and leadership within the immunology community. So I request Professor Dr. Ghazala Jafri to kindly come on. Our second chair for this session is Dr. Dawood Ahmed. He will be joining us shortly. Dr. Dawood Ahmed is an associate professor of Bahria University of Karachi. He did his MBBS from Zakaria University Multan in 1996. He obtained his FCPS in immunology in 2011. He has more than 20 research publications. He is the current president of Pakistan Society of Immunology. His extensive knowledge and expertise in immunology make an valuable asset in advancing our society. So, sir will be joining us shortly. In this session, we have got 3 invited speakers and 4 fee papers. So, I request Dr. Ghazala Jafri to please continue. Thank you, ma'am.
Bismillahirrahmanirrahim. We are going to start the session. Our first speaker is Dr. Rabia Hussain. Because of her health reasons, she has not been able to come. So, recording, please. Her presentation will be shown as a recording. I'll briefly mention the achievements of Dr. Rabia Hussain. She was professor and former head of Department of Microbiology at Khan University, Karachi, and also Professor Emeritus at AKU. She obtained her BSc in 1966, MSc from Karachi University, and PhD from University of Western Ontario, Canada. She's a fellow of the Royal College of Pathologists, and she has many achievements to her. She has done a lot of outstanding work on immunology of microbacterial diseases, and she received the World Academy of Sciences Prize in Medical Sciences, Research Excellence Award, AKU National Honors, Tabgha Imtiaz and Sitara Imtiaz. She's ATC National Distinguished Professor and a fellow of the Pakistan Academy of Sciences, the World Academy of Sciences, Islamic World Academy of Sciences, member of BSI, and the International Society of Infectious Diseases. Yesterday, she was honored by the Lifetime Achievement Award of the Pakistan Association of Pathology. She is patron-in-chief of the Pakistan Society of Immunology. We all pray for her health so she can join us, inshallah, in one of our future activities. So, presentation of Professor Rabia Hussain.
Assalamu alaikum. I would like to start by thanking the Pakistan Society of Immunology for inviting me to give this talk. Please accept my apologies for not being there in person. I will miss meeting friends and colleagues and hearing from them. The focus of today's talk will be to identify the determinants of COVID-19 mortality, and morbidity. First, I would like to share with you some definitions which I know you are all familiar with. But to be on the same page, I have defined them again. Morbidity is the cumulative number of confirmed cases due to a disease. Mortality, cumulative number of deaths in confirmed cases. Incidence of disease is the number of confirmed cases per 100,000 population at a defined time. Case fatality rate is the number of deaths per 100,000 population and is usually used to define trends in case fatality. And disease burden is a cumulative number of confirmed cases at any one time, and this is usually used for high disease burdens and especially for chronic disease burden. This slide depicts the global maps generated by WHO for incidence of COVID-19 in the first week and the mortality at the end of the pandemic. It is obvious that mortality was much lower in countries which are associated with high disease burden and are globally in the lower socioeconomic strata. My talk will address this puzzling inverse relationship between burden of disease and COVID-19 morbidity and mortality. This slide shows a selected number of countries which showed the greatest variability in CFR. What is depicted here is a timeline for CFR from the early phase of the pandemic to the end of the pandemic. The variability ranged between 2 to 15% between these selected countries, with Pakistan showing 2% mortality. It was interesting to note that during the pre-vaccination phase, discrepancy was the highest. The second important point to note is the sharp decline in CFR before any intervention. In Pakistan, this plateau was achieved 1 year earlier than vaccine intervention. So the early period was probably the determining phase for eventual CFR outcome. One question that kept coming up was why there was higher mortality in India compared to Pakistan. This slide therefore compares CFR between Pakistan and India. So perceptions can be deceptive. The differences between India and Pakistan in the initial phase was only on the average 1 to 2% higher, and the trends reversed with India showing lower CFR compared to Pakistan even before vaccine intervention. Again, this data demonstrates that the variability in the early phase was due to some protective mechanisms which helped lower the CFR in these countries, reinforcing the earlier observation that countries with high disease burden show lower CFR. The issues I would like to address in this presentation is one, to understand the link between disease burden and COVID-19 mortality, and secondly, to identify immune mechanisms that may be protective of the first checkpoint. We next compare the global disease burden and COVID-19 mortality. These maps are from WHO dashboard and WHO reports. The intensity of color relates to the disease burden. Total burden of disease in 2019, just before the COVID pandemic, is shown on the left panel, and the COVID mortality at the end of pandemic on the right panel. The countries that stand out are Africa and Southeast Asia, which have the highest disease burden and the lowest CFR. Next, I selected 4 WHO regions for comparison: Europe, Eastern Mediterranean countries, Southeast Asia, and Africa, and compared the chronic disease burden with COVID CFR. Chronic disease burden is shown in orange and COVID in blue. A picture is worth 1,000 words. Countries with the highest chronic disease burden have the lowest CFR. This slide lists some of the most prevalent chronic parasitic diseases in these regions: malaria, tuberculosis, schistosomiasis, and soil-transmitted helminthiasis. We further assess the impact of each of these diseases with COVID-19 CFR. This slide shows percentages of global burden of chronic diseases in comparison with COVID CFR in these 4 regions. Africa carries the highest burden of malaria and schistosomiasis, while Southeast Asia has the highest burden of tuberculosis and soil-transmitted disease. However, the impact of COVID CFR was very similar. and the common denominator seems to be the total burden of disease as shown on the extreme left-hand column. We were interested in the impact of tuberculosis of COVID CFR, as Pakistan has a very high TB burden, ranking 5th in the 30 high TB burden countries according to WHO report. These are the global maps of burden of TB and COVID CFR with the intensity of color indicating increasing burden of disease. The left panel shows global burden of TB and right panel shows COVID CFR. Again, the slides makes the point that TB burden is inversely related to COVID CFR. So the obvious question is, what is the basis of protection against COVID in these countries? Mycobacterium tuberculosis is a slow-growing bacteria with a doubling time of 11 days, and therefore initial immune modulation takes place in the absence of detectable bacterial load, particularly in an early infection due to community exposure and latent infections. However, immune modulation is occurring in these infected individuals and detectable by in vivo and in vitro tests. All countries with high TB burden countries have an active BCG vaccination program in place. BCG is the cross-reactive mycobacteria which showed protection against TB in school children in UK. On this basis, BCG vaccination was introduced in 1960 and is given at birth to date with 95% coverage in Pakistan. Despite this wide coverage in 2020, Pakistan was ranked 5th in the high TB burden countries, showing the inefficacy of BCG vaccine against the transmissible form of tuberculosis. So why has this BCG vaccine been effective? So we come to the next question. Does BCG protect against other non-related infections? It was recognized very early when field trials for BCG were carried out in India and Malawi that BCG not only protected children against disseminated TB, but was also protected against leprosy. Another important observation that was made in field trials was that BCG vaccination at birth reduced childhood mortality. This was shown in Bangladesh, India, Papua New Guinea, and Malawi in Africa. Most of the childhood mortality is associated with respiratory viral infection. This brings us to the next question. Children do not have a mature adaptive arm, which only attains maturity in 14 plus years. So it has to be the innate arm that is providing the protection in early childhood. The two most critical cells here are phagocytic cells, which are part of the mononuclear series of cells, and natural killer cells. Innate cellular system has all the right ingredients for providing cross protection in the first checkpoint. It is fully functional at birth, responds within 0 to 4 hours. Cells recognize epitopes with broad specificity shared across pathogens. Innate system is therefore capable of providing cross protection against a range of pathogens. Macrophage phagocytize pathogens and kill them intracellularly by various mechanisms shown at the bottom of the slide. Natural killer cells can directly interact with infected cells and kill the target cells. All these functions are activated in an orchestrated manner to decrease the pathogen load at this first checkpoint. So why was innate immunity ignored for so long by immunologists? The dogma was that these cells do not have memory and therefore you cannot boost innate systems. This concept was challenged by Nietzsche and his colleagues in 2011 when they demonstrated recall responses to BCG in innate cells, and a new term was coined trained immunity. What they showed was trained immunity was associated with long-term epigenetic and metabolic reprogramming of the innate network such as macrophages and natural killer cells, which showed heightened recall responses to BCG challenge. A role for trained immunity was then extended to BCG protection against viral infections, both in experimental models and in BCG vaccinated children. Najia, other than I, have been long interested in this non-specific immunity of BCG vaccine because of our work in mycobacterial diseases. And in fact, we wrote a review which was published in 2014 on this very topic. In 2014, Najee applied for an 80UC money grant for a pilot study to address the following issues. All the previous studies had focused on early childhood protection by BCG. What we wanted to address were the following issues. Does BCG induce PI decline after the adaptive immune system reaches maturity? Can PI have a role in protection in acute viral infections? Just to remind you that this study was initiated pre-COVID pandemic. The subjects recruited at a mean age of 28 and were initially stratified on the basis of scar presence, which is considered a surrogate marker for BCG vaccination status. Two groups differed on the basis of history of TB contact, which was observed only in 2 out of 8 BCG scar positive groups. There were no other differences in the two groups, including monopositivity. This slide shows the innate cells and cytokines secreted in response to BCG. We used a short-term stimulated 12-hour whole blood culture as this would only pick up early innate responses. LPS was used as a control antigen as this is predominantly a macrophage stimulator. In adults, there were no significant differences in cytokines in response to BCG in the scar positive and negative groups, including interferon gamma, which is considered a surrogate marker for BCG vaccination immunity. All cytokines were uniformly activated in the groups, indicating recall responses to BCG. So the two groups were not stratified for further analysis. To compare the magnitude of recall responses, cytokines were analyzed as signal to noise ratio. So the ratio shown here are the cytokines secreted post-BCG stimulation after deducting cytokines secreted in the absence of BCG stimulation. The highest recall was observed for pro-inflammatory cytokines, interferon gamma and TNF-alpha, which are contributed by both macrophages and NK cells, followed by IL-10, which is contributed by only macrophages. IL-10 is a regulatory cytokine and keeps the pro-inflammatory cytokines in check. Cytokines associated with the T-cell lineage showed non-remarkable activation. So recall responses are associated with the two critical innate cells, macrophages and NK cells, required for early clearance. To determine the specificity of the BCG stimulation, we compared responses of BCG with LPS, which is a known molecule shared across a broad range of pathogens and is a potent selective stimulator of the immune system. BCG and LPS gave similar responses for macrophages secreted cytokines, indicating that the innate network was being stimulated by conserved cross-reactive epitopes in BCG and may be the basis of cross-protection. Such responses activated early would play a determining role in early protection for effective pathogen clearance. The next question was if BCG can induce cross-reactive antibodies as well. Previous evidence by us and others showed that in endemic population, basal levels of IgG are much higher, suggesting an expansion of B-cell repertoire. We have also published an interesting observation in leprosy that there is an expansion of IgG antibodies to a non-relevant pathogen T. gondii in leprosy patients with no symptoms of toxoplasmosis. Our study took longer than we had anticipated, and by this time the COVID-19 pandemic was in full swing. We therefore decided to look for COVID-19 cross-reactive antibodies in our BCG vaccinated pre-pandemic donors. We detected high levels of IgG anti-RBD antibodies in our donors. What we also showed was a high degree of cross-reactivity with BCG, as BCG could block more than 90% of IgG anti-RBD binding. These results strongly suggest that it is the conserved epitopes in BCG that are inducing these cross-reactive IgG anti-RBD antibodies. The importance of these antibodies is that they facilitate the entry of pathogen in a phagocytic cell at the early stages of infection. The identity of epitopes being recognized in pre-pandemic serum remains to be determined. Such information will be very useful in formulation of next generation of vaccines. The puzzling question was why countries with BCG coverage do not show the same level of data protection. This slide provides an answer. Lahore has equivalent BCG coverage but showed much higher rates of COVID mortality compared to high disease burden countries. So again, burden of disease seems to be a key determinant in reducing COVID CFR. This would suggest that in endemic countries, trained immunity is maintained due to continued community. So the overall country data on infectious disease burden may be a major determinant of decreased COVID CFR. Trained immunity is maintained well beyond early childhood due to community exposure. The major role of trained immunity may be early decrease in viral load in COVID-19 infections. I would like to end my talk with acknowledgement to my colleagues, Dr. Najia Talat and Dr. Tara Hassan in the Aga Khan University. Thank you everyone for your attention.
Next I invite Dr.
क्वेश्चन है
नहीं अगर इजाजत देंगे तो
Okay.
Thank you very much. What a nice talk. Thank you, Dr. Nazir, Professor Rafi. Thank you very much. And we always thought that the innate immunity was most important in reducing the viral load. In fact, the non-specific markers and the interferons, they reduce the invaders. So the concept of having the total disease burden in terms of infectious disease or malaria or helminths infection, stimulating the production of interferon to perhaps a higher base level. That could be another demonstration in the population. What happens in the exposed population, what happens in the non-exposed population, say living in relatively better environment, what were the rates, what was the CFR in this and that would be an additional laboratory. So that has been so important, and even in some of the immunopathogenesis, this was demonstrated that interferon levels, in fact the interferon delta came into view, another one, alpha, beta, gamma, and then delta came. So all of those are associated with reducing the invaders. If you have reduced invaders, there would be less chaos in immune response, and the chaos was responsible for, not very well directed immune response. The immune system was overwhelmed with a lot of invaders. So it was not very well directed. That was the main cause of morbidity and mortality. So they were thinking in such a nice, right direction when they worked with the disease burden, one of the perhaps main causes of having increased level of this non-specific immunity in our population. One of the reasons, perhaps, for the lower CFR and rest of the things. So where did this idea originate with your?
Yeah. So basically, actually, I after finishing my PhD, I joined for my postdoctoral, and there was my PI was working on the effect of BCG on melanoma cancer. So that idea came from that BCG has a role in protection against non-communicable disease, and also there are a lot of evidence that suggests that BCG has a role in reducing cancer. And this study was done in bladder cancer, and some of the clinical trials showed efficacy of BCG vaccine in the bladder cancer and also in the melanoma cancer. So when I came back from my postdoc, then I wrote this grant to see whether BCG has an effect on non-specific immunity, and we enrolled only healthy controls in this cohort and not any TB patient or anything. So we basically wanted to see the effect, how innate immune response is stimulating by BCG.
Just a small observation. I guess one of the objectives was that just to see the difference of mortality or disease burden in Pakistan as compared to India as well. So my query is regarding those infections which you thought that these are more prevalent in Pakistan, like malaria, tuberculosis, and so on. Are they not the same in percentage in India as well? Because as far as hygiene and their genetic maybe is almost similar. So does it answer your question?
I think Dr. Rabi also said that this is not clear that why we have higher mortality in India, whether they are, and at the same time they have the same coverage of BCG vaccine. It is not yet clear. Yeah. So it's again come to the your body immune response, how you treat the virus in the first initial phase of infection, which is the first week of infection. So how innate immune response kicks in to eradicate virus. I don't know. We did well with COVID-19, alhamdulillah, but not in.
Thank you.
Our next speaker is Dr. Najia Talat Iqbal. She is an associate professor at Department of Biological and Biomedical Sciences and vice chair of Research Department of Pediatrics and Child Health at Aga Khan University. She did her PhD in Immunology in 2010 from University of Karachi. She did her postdoctoral training in John Vine Cancer Institute, USA in 2011. She had more than 50 research publications. Now I invite Dr. Najia Iqbal.
Assalamu alaikum. Dr. Rabia's talk has set the stage for me for COVID-19, so I'm not going into the details of COVID burden. So the talk is a little bit related to what she said, but this is about the long COVID. So the title is to exploring geographic variation in the clinical presentation of long COVID, implication for phenotype association in Brazil and Pakistan. So this is basically a multi-site project. I will give you detail of this project in a while. So what is post-acute sequel of long COVID, which is also known as PAS? PAS, long COVID, long haul of COVID, these are all synonyms. These are abbreviations which are usually used for long COVID. So PAS basically refers to the persistent symptoms experienced by an individual after recovering from acute phase of long COVID. And I think some of you in this hall experiencing the same thing. And I think most of us are going through this long haul of COVID or long sequel of COVID in many forms, like the symptoms as shown here is a brain fog, memory loss, anxiety, joint pain, muscle pain, sleep disturbance, and fatigue. Majority of us are not doing as well as we were doing before pandemic. So alhamdulillah, pandemic has ended, but I think the long COVID situation is still going on, is still prevailing in our society. So, but in US, the ratio is very high. I don't know whether they have invested a lot in studying long COVID, but they say that in in the five Americans, they are suffering from COVID. So the ratio is very high. I don't know what is the percentage here because we have not invested much in understanding the long COVID prevalence in Pakistan. So, but in US, is one in five individual is suffering from any symptom of the long COVID which prevails, which limited their activity, which limited their life, work life, and absenteeism from the work, and these are several things which have been seen in US. So they have invested a lot in understanding this, and this is the Recover study which recruited over the 9,000 participants, and which is basically the title is Researching COVID to Enhance Recovery. And this trial has shown that there are 37 symptoms across multiple pathophysiology which are seen or observed in patients with long COVID over the period of 6 months after the infection. So if you have the infection, if any symptom persists from the initial infection, or you develop any other new symptom after 28 days or 1 month of the initial infection, this will be regarded as a long COVID. So the key question they address is what symptoms are differentially presented in individuals 6 months or more after infection compared to uninfected individual, and what are those 37 symptoms which are present across the multiple pathophysiology. The problem is that there is no framework or definition of the PAS. So if you look at the literature, some people would say that okay, more than 1 month post-infection is long COVID. Some say no, it is more than 6 months post-infection after long COVID. So there is no framework which can be used and generalized across the sites. So we were also part of this big network which Nida has shown, and we were basically funded for studying this long COVID. Just to give you an overview that the long COVID has multiple early factors which anticipate long COVID, and this study was published in 2022, and they basically recruited 2 cohorts. One is INCOV cohorts and other is Harvey cohorts. Harvey is basically hospitalized patient and INCOV are ambulatory patients. And they followed these patients for over 1 to 2 weeks or up to 2 to 3 months. And what they found is that their symptoms of the PAS, as I identified, as a gastrointestinal symptoms, cough, neurological symptom, these were highly correlated with ANA antibodies, anti-nuclear antibody, as you can see in the second graph here. In the second graph here, you can see they are highly correlated. And in the second graph, you can see there is a hyperinflammatory state in the long COVID in these patients. And this hyperinflammatory state goes up to T3 times. Like it remains the inflammation is so high that it continues up to several months after long COVID or after COVID. And you can see these are high correlation you can see with the CXCL10, CCL, all chemokines and other inflammatory biomarkers like interferon alpha 2 was highly correlated with other chemokines. And you can see there are a number of anti-nuclear antibody were also associated with this plasma cytokine and plasma chemokine. So this basically shows that in long COVID there is a hyperinflammatory state which possibly cause endothelial damage or which may cause some kind of inflammation or tissue damage, which basically persists over time and which cause these symptoms, which may be neurological symptoms, which could be physical symptoms like fatigue, exertional malaise, like people say that they can't climb the stairs now. They don't do gym as regularly as they used to do it. So these were the things that were associated, possibly due to uncontrolled inflammation that is going on. It could be related to the viral persistence. So we think that viral has shed off from our body, but studies have shown that there are viral particle is still present in GI tract, and they have in some studies they have found out that there are viral particles present in the feces as well. So we, as a part of United World of Antiviral Research Network, we initiated this study, and there were 2 cohorts, one from in Pakistan and another from Brazil. And we basically interested to see that the phenotypes of the long COVID are similar across Pakistan and Brazil, or they are different, because we have a different genetic makeup, we have different lifestyle and other features. So the clinical case definition of PAS is that somebody have continuous symptomatic COVID-19 4 to 12 weeks, or it goes beyond 12 weeks. So this is the definition. It comes under the long COVID. New symptom arise or persist for longer than 4 weeks. after the initial infection. So we included all the individuals greater than 18 years of age infected with SARS-CoV within last 3 years and presenting with a wide range of physical and mental consequences that persist for 4 or more weeks after SARS-CoV infection. And PAS control 1 are the asymptomatic person who was previously infected with SARS-CoV and was the household contact of the index case. And the second control is anybody from the community. So this is our study design. This is the prospective and the longitudinal prospective study design, and also the retrospective study design. And we enrolled, so far we have enrolled 32 cases and 26 control. This, we have a structured questionnaire in which we collect information about demographic, diagnosis, comorbidities, medication, COVID vaccine, and the follow-up. And we followed them at different time points. And here you can see the routine tests we do. And this routine test includes CBC, ESR, lipid profile, and other tests. And then for the research test, we collect 3 samples over the period of 12 months, which is basically estimation of the some biomarkers in the fecal and the in blood samples. So the aim was to characterize spectrum of clinical symptoms and distinct phenotype of long COVID, and to understand the phenotypic differences across the geographic Pakistan and Brazil, and to understand whether there is any association of immunological parameters with the long COVID symptoms. So first, gender. So here you can see that it was expected, and you know in the literature also suggests that females are at more risk. There is a difference when COVID hit male gender in terms of case fatality rate. But in terms of long COVID, females are somehow more at risk for long COVID. And this was consistent with Brazil as well. You can see the 82% were female in the long COVID. So this is same across the board. And then we categorize COVID into short term, intermediate, and the long term. And here you can see our enrollment was, most of the enrollment was between 2021 and 2022. And the majority of our patient 87 have more than 6 months post-infection. As compared to the age, you can see that in Pakistan, more younger people are affected by long COVID. But in Brazil, you can see more of this, more than 60 years people are affected. So there is a difference in the age category between Pakistan and Brazil. Also in terms of symptoms, we compare this on the table. You can see that topmost is the fatigue in Pakistan, but in the topmost in Brazil is joint pain. And here you can see the graph which shows that 90% of our people are suffering from the fatigue, breathlessness, and weakness that limits their activity. And this is the overall comparison and the comparison of the Pakistan and Brazil. So in terms of memory loss, we wanted to see whether this memory loss is associated with the younger age or the older age. So we categorized into less than 40 and more than 40. And we found that the more than 40 years showed that they mentioned that they had some kind of memory difficulty in recalling, or they have a brain fog or something like that. Then we have a very detailed questionnaire, and one question was that have you fully recovered from COVID or you are not recovered? So 87% said that we have not recovered from COVID completely. And on the base of that questionnaire, we have certain questions that we ask that do you feel fatigue? So they said 78% of people said that yes, they have fatigue, some kind of fatigue during their routine course. And 56% responded that they have some kind of weakness that limits their daily activity, and they showed that they are not fully recovered. Here then we compared what is the immune parameters, and this is a very preliminary analysis, and we found that this not the lymphocyte, neutrophils are associated with these symptoms like fatigue and the cough, and here you can see muscle ache. And also we found that the fatigue was also related to a lot of other COVID symptoms, loss of smell and taste and other things. And then we looked at particularly the neutrophils, lymphocyte, monocyte, ESR, and CRP. And then we found that those who had responded yes for the fatigue, they had higher neutrophil count and higher ESR and slightly raised CRP for the weakness here. You can see the CRP was slightly raised, and then they have again the neutrophil is somehow coming up as a biomarker for the fatigue and not the lymphocyte. Similarly for the memory loss and the breathlessness. Some people responded that they had headache, severe headache, or the frequent headache coming and going, and they also showed that they had lymphocyte count a little bit higher, but not other parameters. So in conclusion, we found that the female gender was predominant in both cohorts, and Asian cohort had more younger people who were somehow affected with PAS and not the older one, as we saw in the Brazil. Common symptoms include fatigue, breathlessness, and weakness, which were the three major symptoms that we found, and no significant difference was observed in the lab parameter in PAS symptoms, positive and negative. Autoantibodies are the one way to understand we are working. We have not started, but this is our plan to do the autoantibodies against chemokine and the cytokines in these patients to see whether they have these prolonged autoinflammation in their body. And also we are looking at the fecal shedding of SARS-CoV virus in the fecal sample. We are also collecting their fecal sample. So this will also show that if there is a persistence of viral in those people. And this is our principal investigator, Isadora, from Brazil, and her team. And this is my team, and I really want to acknowledge the UWAR network and the funding from NIH for this UO1 grant. And also this is the team, and these are three infectious disease doctors who basically see these patients in their clinic regularly at the interval of 3 months. And we have these clinical labs and other things done, and doctors basically check them for every follow-up. So, the end, thank you. And
The forum is open for any questions.
Assalamu alaikum. Thank you, Dr. Najia, for highlighting a very important topic. I just want to ask that have you compared the hematological markers with the healthy controls or not? Because there is a higher trend of neutrophils, and sometimes lymphocytes are also reported that they are higher in long-COVID. But have you compared them? Is there any significant difference between these hematological markers of healthy controls and these long-COVID patients?
yes, so we are doing these labs for healthy controls as well, but the data was not entered in the red cap. For the analysis, it was not QC. So Kumail and Nida, they are. So yeah, this is our plan of comparing both COVID, PAPs, and the control 1 and control 2, and see how their labs are doing.
Thank you very much. Is there any relationship between the severity of disease and the long COVID, and in the lab, has anyone looked at the long-term activation markers on lymphocytes, say HLA-DR expression, on say helper and suppressor, or the subsets? The different readings there, are there any of those studies?
I think this is a very valid question. Unfortunately, we are not doing flow cytometry, but we are keeping the samples for RNA transcriptomics. And the RNA transcriptomics will show some of these signals. What are those immune parameters or immune cells are upregulated in the long COVID, and what are those are downregulated. So this is something that in pipeline that we'll do some RNA-seq analysis.
And the severity of disease and association with.
So yeah, so we collect data about whether you were hospitalized during COVID, whether you had any, you were on ventilation, did you have any assisted oxygen? So that questions were included in. So I think that in the analysis we can put in those as a terms of severity. But unfortunately, fortunately we are not seeing COVID cases hospitalized right now. So we, for prospective analysis, I don't think we can do, but for the retrospective, we can collect this data.
Dr. Nadia, thank you very much for such an interesting talk. On a lighter note, Alhamdulillah, I never got COVID. But I have all these 35 symptoms. So, is there a possibility that I had subclinical infection at any time?
Yeah.
And I think, as Brigadier had just said, one of such markers might able to sort my issue.
Yeah.
I thought
Same.
this was
Yeah, this-
my description.
Yeah. So,
Is it
so
age
same thing.
or
Yes.
is it really
No,
age or?
I am young enough, sorry.
So same thing is
No,
that,
no,
that
no.
you must have. So it is unlikely. I never got my PCR test positive for COVID-19 during the pandemic. I had multiple PCR done, but it never came positive. But the thing is that my innate system was somehow clearing virus, and it was not coming. But you know, the virus persistence in the body we cannot rule out unless you do the maybe it is in your GI tract or maybe somewhere. So like I mean, some kind of inflammation, you go through this. Even during the innate immune response, you will go through this inflammatory response for clearing of the virus. So I don't know, and how your body is reacting to a new invader, that is a thing to debate on. Maybe your body may have some take it very light, and some immune response are very heightened, like a hyperinflammation kind of state. So that depends from person to person.
Could it be due to repeated vaccination? Because I've been vaccinated 6 times.
Yes, this could be due to. And I personally, I feel I'm not sure, I've not studied, but personally, when I had this Pfizer, I had a lot of issues with fatigue and breathlessness and exertion. This happened not with Sinopharm, but after Pfizer, I had this.
The threshold for these symptoms, the vaccination is set at 7 vaccines, not 6. So you had 6.
I had 6.
So
I
7
had
is the threshold.
I had
Not
the
related.
so I had the new variant wali in
At least 3 of them.
So I have a question in line with Mr. Ghazala. Did you check antibody titers also in these patients?
Yes, so we check for a spike protein. We check for both PAS, and the problem is that now we are enrolling at this point where everybody is vaccinated. So it's very hard to find. We kept the enrollment criteria that control should be spike negative, but unfortunately we can't find any. So now we have switched it to have a nasal swab, negative nasal swab for the community controls and the controls, and also the fecal negative fecal shedding of virus as a control.
And for the NP also, I mean, you can have some kind of segregation if they had the infection, maybe subclinical infection, or they were just vaccinated. Because if they are positive for NP,
Nuclear protein? No, not for the. We have not checked for nuclear. We check for spike protein. But we can check for nuclear. Nuclear, we have our own ELISA we can do. But yeah, it's very hard to find at this moment anybody who's spike negative, because everybody is vaccinated, or I don't know.
Different combination of vaccines have been given.
Yeah.
There is another reason.
Yes, it could be due to this.
and post-viral syndrome is another factor.
So post-viral syndrome is everywhere. In dengue also you see post-viral syndrome. But the COVID, I think people had invested a lot in studying long COVID, and that's why I think there were more studies and more papers, but not for dengue, not for other viral infection. But I think there is post-viral sequel for every virus. Thank you.
Thank you, Dr. Najia. Our next speaker is Dr. Fahim Shehzad Khan. Dr. Fahim Shehzad Khan, a lecturer at University of Health Sciences, Lahore. He has a strong academic background, completed a bachelor degree in clinical laboratory sciences and master in microbiology. He has also earned an MPhil and PhD in immunology. Dr. Fahim has served as and American Society for Microbiology Young Ambassador to Pakistan, and he has over 35 research publications in immunology. I invite Dr. Fahim.
थैंक यू सो मच सर। यहाँ पे मेरे तमाम मेंटोर्स और टीचर्स बैठे हुए हैं, तो मुझे बहुत ही फीलिंग हो रही थी, इतना लंबा आपने मेरा इंट्रोडक्शन कराया। थैंक यू सो मच फॉर दैट। एंड फर्स्ट ऑफ ऑल आई वुड लाइक टू एक्नॉलेज ऑल माय टीचर्स एंड मेंटोर्स for training me in a such a way that I am today here standing and having some sharing my thoughts here. So today I am here to talk about artificial intelligence, its role, challenges, applications, and future directions in clinical immunology. आप सबने पिछले साल से बहुत इंफॉर्मेशन आपके पास होगी इसकी आर्टिफिशियल इंटेलिजेंस की, और फ्रॉम 2022 जब चैट जीटीपी और इस तरह के दूसरे सॉफ्टवेयर्स रिलीज हुए, तो इससे पूरी दुनिया के अंदर एक तरह का मतलब इंफॉर्मेशन और बहुत सारी चीज़ें ऐसी हैं जिससे लोगों के अंदर उनकी लाइफ भी बहुत ज़्यादा इजी हुई। और कुछ लोगों का ये कंसर्न था कि आर्टिफिशियल इंटेलिजेंस जो है बेसिकली एक टाइम आएगा जब इंसानों की अहमियत ख़त्म हो जाएगी और एवरीथिंग विल बी डूइंग मशीन्स। सो जहाँ पे हंड्रेड्स ऑफ पीपल काम करते थे कंपनीज के अंदर, वहाँ पे एक पर्सन बैठा होगा और सारा काम जो है ऑटोमेटिकली हो रहा होगा। बहरहाल वी आर टुडे हियर कि हम आर्टिफिशियल इंटेलिजेंस को अपनी रूटीन जो हमारी फील्ड है उसके अंदर किस तरह यूटिलाइज कर सकते हैं। सो आई शुड स्टार्ट हियर। सो बेसिकली व्हाट इज आर्टिफिशियल इंटेलिजेंस? बेसिकली AI refers to computer systems that are capable of tasks needing human intelligence. कि वो तमाम काम जो कि इंसान कर सकता था, अब वो तमाम हम कंप्यूटर के थ्रू भी कर सकते हैं। It simulates human cognitive processes like learning, problem solving, and decision making. In computer science, basically AI specializes in making intelligent machines that learn, adapt to new information, and perform tasks such as recognizing images, understanding speech, making decisions, and translating languages. अगर हम टाइप्स की बात करें AI की, तो अभी तक जिस लेवल पे हैं दैट इज नैरो आई। इसकी 3 कैटेगरीज हैं। जितनी भी अभी तक इसमें तरक्की हुई है तो अभी दैट फॉल्स इन नैरो AI। तो उसके लिहाज से अगर आप सोचें, जो इसकी 3rd जनरेशन है दैट इज सुपर AI। तो अभी जो हमारी अगर ह्यूमंस के लिहाज से जो हमारी एफिशिएंसी है उसके लिहाज से देखें, तो जो करंट नैरो AI है it is beyond the capabilities of human. So when general AI and super AI जब आ जाएंगे, ठीक है, जिसके अंदर अगर आप super AI में देखें तो system where the intelligence of a machine surpasses all forms of human intelligence in all aspects and outperforms human in every function. ठीक है, तो उस वक्त हो सकता है कि जो ह्यूमंस हैं उनको अपनी जो एफिशिएंसीज हैं उन पे बहुत ज़्यादा काम करना पड़ेगा, अदरवाइज उनकी जो यूटिलिटी है इन फ्रंट ऑफ दोज मशीन्स वो फिर बहुत ज़्यादा नैरो हो जाएगी। मेडिसिन की अगर हम बात करें तो AI hold significant promises in revolution in disease diagnostics, treatment, और तमाम जो ब्रांचेस हैं जिसमें डायग्नोसिस की अगर हम बात करें वहाँ पे इसकी बहुत ज़्यादा यूटिलिटी है। ट्रीटमेंट्स और प्रिवेंटिव जो स्ट्रेटजीज हैं उसके अंदर बहुत ज़्यादा इसको यूज़ किया जा रहा है थ्रू द वर्ल्ड। AI algorithms possesses the ability to analyze extensive medical databases, recognize patterns and offer valuable insights. Basically AI के अंदर एक सेट ऑफ इंस्ट्रक्शंस हम जो है यूज़ करते हैं और जो भी डेटाबेसेस हम उसको प्रोवाइड करते हैं प्रीवियसली उन डेटाबेसेस को यूज़ करते हुए और जो इंस्ट्रक्शंस प्रोवाइड की जाती हैं उनकी बेसिस पे वो आपको डिसीजंस करके देते हैं। से फॉर एग्जांपल अगर आपके पास पेशेंट्स का डेटा है लास्ट 10 इयर्स का किसी भी इम्यूनोलॉजिकल डिसऑर्डर की अगर आप ले लें, अब आपने कोई टेस्ट करवाया, उसकी आप इंटरप्रिटेशन करवाना चाह रहे हैं, आपके पास टाइम नहीं है, ना ही आपके पास इतना स्टाफ है जो कि उसको इंटरप्रिटेट कर सके। आप जस्ट क्या कहेंगे कि वो जो रिजल्ट्स आए हैं आप वो शेयर कर देंगे इसके साथ, वो आपने जो इंस्ट्रक्शंस ऑलरेडी दी हुई हैं और जो डेटाबेसेस प्रीवियसली अवेलेबल हैं उनको यूटिलाइज करते हुए आपको परफेक्टली इंटरप्रिटेशन देगा जो कि एक नॉर्मल ह्यूमन हो सकता है उस तरह से ना दे सके। तो इसकी यूटिलिटी जो है डायग्नोस्टिक्स के अंदर और स्पेशलाइज्ड जो ट्रीटमेंट्स हैं, छोटी छोटी चीज़ें जो कि ह्यूमन नॉर्मली जब आपके पास पेशेंट आता है आप उसको देख रहे होते हैं तो हो सकता है हम उस चीज़ को मिस कर देते हैं, बट जो एआई के अंदर ये एबिलिटी है कि वो छोटी पिन पॉइंटेड जो चीज़ें हैं उनको भी वो यूटिलाइज कर सकता है। This technology assists healthcare professionals in making more informed decisions and creating personalized treatment plans. Integration of AI in healthcare also aims to minimize medical errors, ultimately enhancing patient outcomes. इम्यूनोलॉजी के साथ इसकी अगर हम लिंकेज की बात करें तो AI aids in analyzing extensive data sets, detecting patterns, making predictions, assisting in novel discoveries and treatment approaches in immunology. Potential revolution in clinical immunology. इसमें the integration of AI and immunology holds promise in transforming clinical immunology, facilitating creation of personalized therapies, diagnostics and vaccines. करंट एप्लीकेशन की अगर बात करें जो कि वर्ल्ड वाइड यूज हो रही हैं तो इसमें इनहांस डिजीज प्रिडिक्शन एंड डायग्नोसिस में इसको यूज किया जा रहा है, पेशेंट स्पेसिफिक ट्रीटमेंट्स में इसको यूज किया जा रहा है एंड एक्सेलरेटेड ड्रग डिस्कवरीज में जो है आर्टिफिशियल इंटेलिजेंस को यूज किया जा रहा है। कुछ मैं एग्जांपल्स आप उनसे शेयर करूंगा लास्ट फ्यू इयर्स में जो कि एआई को यूज करते हुए जो काम हुआ है। इसमें सबसे पहले जिस तरह कोविड के भी हमने 2 टॉक्स सुनी है उसके अंदर वैक्सीन की भी बात हो रही थी, तो बेसिकली जो वैक्सीन का प्रोसेस था उसमें एआई का बड़ा रोल था कि बड़ा हमने बहुत इजीली एक इतनी आसानी के साथ एक वैक्सीन हमने तैयार कर ली विद लिमिटेड स्पैन ऑफ टाइम। तो उसकी जो बेसिक जो उसके बिहाइंड जो था वो एआई का बड़ा रोल था। सो रिसर्चर्स यूटिलाइज एआई टू डिजाइन अ स्टेबल स्पाइक प्रोटीन ऑफ SARS-CoV-2 crucial for COVID-19 vaccine development. Predictions by AI model enhance stability and immunogenicity validated through experiments. And several vaccines जिसमें नोवैक्स वैक्सीन है आर यूटिलाइजिंग एआई डिजाइन जो है ये करंटली भी इसको यूज किया जा रहा है। एआई फॉर प्राइमरी इम्यूनोडेफिशिएंसी डायग्नोसिस में भी इसको यूज किया जा रहा है एंड एआई मॉडल बाय यूसीएलए डायग्नोज प्राइमरी इम्यूनोडेफिशिएंसी यूजिंग इलेक्ट्रॉनिक हेल्थ रिकॉर्ड डेटा, analyzing clinical notes, laboratory tests and medications achieved a high accuracy of 93% in PAP diagnosis compared to genetic testing. Allergic diseases में अगर इसकी हम बात करें, researchers from Icahn School of Medicine and University of Tartu predicted allergic diseases using microbiome data, analyzing gut and nasal microbiota, achieved an 86% accuracy in predicting allergic diseases, revealing novel insights into immune regulation. There are a few limitations. Of course, क्योंकि अभी ये जो टेक्नोलॉजी है अभी इतनी वेल एडवांस नहीं हुई और अभी इसमें बहुत ज़्यादा काम चल रहा है। सो डाटा क्वालिटी एंड अवेलेबिलिटी, आपको अंदाजा है कि जो डाटा सेट्स इसको चाहिए होते हैं, जो अगर हम इससे जो चीज़ें अचीव करना चाहते हैं लाइक डायग्नोसिस है, ट्रीटमेंट्स है, तो इसके लिए हमें जो डाटा सेट चाहिए तो बहुत लार्ज चाहिए होता है और उसकी जो वैलिडिटी है, उसकी जो क्वालिटी है दैट इज आल्सो वेरी वेरी इंपॉर्टेंट। तो ये जो है इसका लिमिटेशंस हैं। देन इसकी इंटरप्रिटेशन है एंड वैलिडेशन, ये दोनों चीज़ें जो हैं अभी इसकी लिमिटेशंस हैं क्योंकि जब आप लार्ज डेटा यूज़ करते हैं तो उसमें फेक जो डेटा है उसके भी चांसेस होते हैं या फिर मिसलीडिंग जो डेटा है वो भी हो सकता है। उन सारी चीज़ों को रूल आउट करना जो है अभी इसकी नीड है। इसके अलावा एथिकल एंड लीगल इम्प्लीकेशंस इसकी हैं। से फॉर एग्जांपल आप पेशेंट्स का डेटा यूज़ कर रहे हैं फ्रॉम द लास्ट 10 इयर्स और 20 इयर्स, उसमें बहुत सारी इंफॉर्मेशन जो है वो जिसका आपके पास कंसेंट नहीं है, तो वो तमाम चीज़ें कंसेंट के लिहाज से आपके लिए पॉसिबल नहीं है कि लास्ट 10 या 20 इयर्स में जो पेशेंट्स आपके पास हैं उनका डेटा है, अगर आपने उनको उसको यूटिलाइज करना है देन उसको किस तरह आप मेक श्योर करेंगे कि उसकी प्राइवेसी भी हो, उसका कंसेंट भी लिया गया हो, अकाउंटेबिलिटी हो, फेयरनेस हो, जस्टिस हो। तो ये तमाम जो इश्यूज हैं अभी इनको डिस्कस करना और इनको रूल आउट करना ज़रूरी है। Again, advancing AI techniques for immunological data handling. Current AI methods जो available हैं may struggle with capturing the full complexity, dynamics and interaction of immunological data. If we look at our immune system, तो उसके अंदर it is very complex system जिसमें multiple cells interact कर रहे हैं, cytokines हैं, chemokines हैं, different other proteins हैं, तो उन सारी चीज़ों को अंडरस्टैंड करना अभी एआई के अंदर ऐसे मॉडल्स की हमें ज़रूरत है कि ये तमाम चीज़ों को वो अंडरस्टैंड कर सके और उनको यूटिलाइज कर सके। सो एडवांस टेक्निक्स लाइक डीप लर्निंग, नेचुरल लैंग्वेज प्रोसेसिंग, कंप्यूटर विजन, रीइंफोर्समेंट लर्निंग ऑफर्स मोर पावरफुल टूल्स, बट ये जो टूल्स हैं अभी तक अवेलेबल नहीं है, इन पे काम चल रहा है। जैसे ही ये जो है मार्केट के अंदर आ जाएंगे देन फिर इसमें बहुत ज़्यादा मज़ीद हेल्प होगी हमें। इसी तरह इंटीग्रेशन ऑफ डाइवर्स डेटा टाइप्स फॉर होलिस्टिक व्यू, कि ओवरऑल जितने भी प्रोसेसेस हैं उन सबको, जिस तरह एक ह्यूमन पर्सन है या कोई भी पर्सन है वो तो सारी चीज़ें, एक एक्सपर्ट इम्यूनोलॉजिस्ट है उसको तमाम प्रोसेसेस का अंदाजा है, और जब भी वो कोई डिसीजन ले रहे होते हैं उस डिसीजन के बेसिस होती है ऑल द प्रोसेसेस। बट क्योंकि एक मशीन है इसके अंदर जब तक हम एक एक छोटा छोटा पॉइंट भी उसमें ऐड नहीं करेंगे उसका डेटा नहीं होगा, तो उस तरह से एग्जैक्ट जो मॉडल है वो उसका वर्क नहीं कर पाएगा। तो इसमें इंटीग्रेशन फॉर होलिस्टिक व्यू डिमांड्स मोर रोबस्ट मेथड फॉर डेटा फ्यूजन, एनालिसिस एंड इंटरप्रिटेशन टू प्रोवाइड अ कॉम्प्रिहेंसिव पिक्चर ऑफ इम्यून सिस्टम एंड इट्स इंटरैक्शंस। Again, creating AI from frameworks simulating immune system functions. Current AI model जो है might not entirely replicate the complexity, adaptability and evolution of immune system. Building AI frameworks simulating immune systems functions require more sophisticated models that can learn, adapt and evolve realistically in response to various stimuli. So this is also a limitation. इमर्जिंग अगर टेक्नोलॉजीज की हम बात करें एआई में, तो उनमें जो नेक्स्ट टेक्नोलॉजी जिस पे काम हो रहा है दैट इज रीइंफोर्समेंट लर्निंग। बेसिकली रीइंफोर्समेंट लर्निंग क्या है? इट विल लर्न फ्रॉम एक्सपीरियंसेस इन एन एनवायरनमेंट वाया ट्रायल एंड एरर। कि आप एक ऐसी मशीन बनाएंगे या एक ऐसा मॉडल बनाएंगे, अगर वो किसी एनवायरनमेंट में आप उसको छोड़ देते हैं, इट विल लर्न फ्रॉम दैट एनवायरनमेंट एंड यूटिलाइजिंग दैट डेटा देन इट विल मेक डिसीजंस। So this is the future technology जो कि अभी आई नहीं है। Complex problem solving, sequential decision making and natural language processing जो टास्क हैं वो आप reinforcement learning के थ्रू कर सकेंगे। इसी तरह deep learning है, it will utilize multiple layers of neural networks to learn from complex data। इसमें आप अगर देखें कि अगर आप immune system की हम बात करें तो there is a network between proteins like cytokines, chemokines, cellulars। सो इन तमाम के दरमियान में नेटवर्किंग के हर एक को मिला के, तो ये जो डीप लर्निंग टेक्नोलॉजी है इसके अंदर ये एबिलिटी होगी कि इट विल क्रिएट नेटवर्क्स, एंड व्हेन इट विल बी अवेलेबल इन फ्यूचर देन वी विल इजीली कैन यूटिलाइज इट इन इम्यूनोलॉजी एज वेल। And other emerging technology is natural language processing. It will help machines understand, generate and manipulate human language. इसकी एप्लीकेशन जो है इंफॉर्मेशन एक्सट्रैक्शन, सेंटीमेंट एनालिसिस, दैट इज वेरी वेरी इंपॉर्टेंट। कि एक आपके पास पेशेंट आता है और आप उसकी लैंग्वेज से ही वो जो मशीन है इट विल अंडरस्टैंड, इट विल क्रिएट डेटा कि वो क्या चीज़ आपसे एक्सप्रेस करना चाह रहा है, ठीक है। टेक्स्ट समराइजेशंस, मशीन ट्रांसलेशंस कंबाइंड विद जो डीप लर्निंग थी और बाकी जो हमने प्रीवियस टेक्नोलॉजीज उन सबको मिला के जो नेचुरल लैंग्वेज प्रोसेसिंग है उसके थ्रू जो मॉडल्स क्रिएट करेंगे देन इट विल बी वेरी वेरी यूजफुल इन फ्यूचर। अगर हम इसकी एप्लीकेशंस की बात करते हैं क्लिनिकल इम्यूनोलॉजी में, तो देन देयर इज कंप्यूटेशनल वैक्सीन डिजाइन जो कि ऑलरेडी हम डिस्कस कर चुके हैं। देन आप इसको यूटिलाइज कर सकते हैं म्यूटेशन इन्वेस्टिगेशंस, न्यूट्रिशनल इम्यूनोलॉजी एंड देन माइक्रोबायोम एनालिसिस एंड ड्रग डिस्कवरी के अंदर ये तमाम चीजें आप वहाँ पे इसको यूटिलाइज कर सकते हैं। इसके अलावा एक और जो टेक्नोलॉजी है दैट इज ओमिक्स एंड मल्टी ओमिक्स डेटा टेक्नोलॉजी। बेसिकली जिस तरह आप जीनोमिक्स, ट्रांसक्रिप्टोमिक्स, प्रोटियोमिक्स ये उसके अंदर इसको यूटिलाइज करते हैं और इसकी बेसिस पे जो भी आपके डाइवर्स जो ओमिक्स डेटा होता है उसको मोलेकुलर मैकेनिज्म और इम्यून रिस्पांस बायोमार्कर्स को आप यूटिलाइज कर सकते हैं बाय यूजिंग दिस टेक्नोलॉजी। अगर हम इसको समराइज करें, so AI facilitates the handling, integration and simulation of large and intricate immunological data from various technologies like omics, imaging and sensors. Challenges की अगर हम बात करें such as data quality, interpretability है इसकी, validation, ethical consideration and need addressing to leverage AI effectively. Then advanced जो techniques हैं कि हमने बात की utilizing advanced techniques like reinforcement learning, deep learning, and natural language processing can significantly enhance immunological data handling and generation. If I conclude this, AI offers vast opportunities for clinical immunology and other disciplines, fostering better comprehension, intervention, and prediction. It stimulates new discoveries, collaborations, partnerships, fostering innovation in clinical immunology and beyond. AI serves as a tool to augment and amplify human intelligence and creativity rather than replacing or diminishing them. So this is the basically take-home message. Okay, so these are few tools. There are more than 2,000 tools which are available based on AI technologies. Just you can go on OpenAI and then you can find relevant जो tools हैं. इसमें जो मैं use करता हूँ that is the last one, ChatGPT. I mostly use in my daily activities. Like for this presentation, I had most of the help from ChatGPT. So this is very useful. Like if you are a practitioner, you can get information for anything that comes in front of you, and if you don't have any clue, just go on to ChatGPT, ask him that I need your help, and this is my problem. He will come up with many solutions. So you can take up anything that is most relevant to you. Again, ChatGPT freely that is available. It is not current version, that is up to 27 data utilizes. But if you want to have updated data that is currently available, then you can utilize Microsoft जो इनका browser है, उसके अंदर ये chat के option है. If you go and utilize this one, it will give up-to-date informations. It is freely available, and you can use updated ChatGPT 4, and it will come up much more information that may not be available around you. There are many tools available. आप इनमें से कोई भी choose कर सकते हैं. आप अपने daily activities में according to your जो भी आपकी है. This is basically artificial intelligence creator that he shows me like I am this one. I just describe myself to him that I am like this person and make a picture of me. And in 1 second, he just made a picture of me. And just for you people. Yeah. Yeah. You can chat with him and then you can have an actual picture as well. So it was just to have a information that you can do anything with using this technology as a medical professional as well as a scientist and as well as a teacher. Even if you want to make a case study for your class, then you just go and ask him that I want this case study for my paper or something else. It will come up with a very comprehensive case study and with answers for that as well. And even you can use for creating a very useful MCQs, SEQs, each and everything. So, like this is a technology that can make your life easier, but of course you can have your own input in this. With this, thank you so much for giving me this opportunity and listening me. Thank you.
House is open for questions.
Thank you. Please, कोई भी चीज़ करे ना, तो इसको देखिए तो सही में. आपको पता है पाकिस्तान में अभी तक पर्सनल डिजिटल डेटा प्रोटेक्शन के लिए कोई एक्ट नहीं है, सो बिल है. सो यू नीड अ लीगल कवर. जब आपने बात की ना कि पेशेंट रिकॉर्ड्स और सारा कुछ. दूसरा, अगर मैं ये प्रेजेंटेशन कुछ करना चाहूँ एआई की कैपेबिलिटीज डेमोन्स्ट्रेट, आई वुड टेक अ रियल प्रोजेक्ट, Rumeza's project, yeah, Dr. Najia's project, and demonstrate the actual softwares, how I would use them and predict the results, and comparing them to whenever the actual results come out. So that's the second thing. And using these softwares for manuscript preparation and everything, that's not encouraged. What are you talking about? So in fact, they have now included questions that whether you have used this, and they advise editors and reviewers so that to use the software so that they can recognize whether this software has been used or not. So it is not to be projected like like it is a solution for everything. So there are so many reservations. So if you want to demonstrate, use this tool for this demonstration, this tool for this tool for this, and then go through each step of the project, like predicting proteins for prospective candidates for vaccination. So that there should have been number of steps and demonstration and actual software with examples, not a general sort of GPT created presentation. So I'm sorry, but this is not to be done.
Thank you so much, sir, for your insights and inputs. Actually, in the purpose जहाँ पे जो हमने discuss करना था वो यही था कि you have both positive aspects as well as the negative aspects. So जब भी कोई टेक्नोलॉजी आती है तो अभी इसका दुनिया में बहुत ज़्यादा कंसर्न भी है, जैसे आपने हाईलाइट भी किया कि इसमें जो ह्यूमन का जो इनपुट है और उसकी जो यूटिलिटी है उस पर बहुत ज़्यादा क्वेश्चन आएंगे इन फ्यूचर, और इसके लिए हमें लीगलाइज वेज जो है वो अभी अवेलेबल नहीं है, उनको यूटिलाइज करना चाहिए, क्योंकि यहाँ पे फॉर दिस प्रेजेंटेशन एक्चुअली इसमें जो भी आप सॉफ्टवेयर यूज़ करते हैं उसमें इनपुट या थॉट्स आपके अपने होते हैं, जस्ट आप इसको यूज़ करते हैं। बट हैविंग सेड दैट, क्योंकि टाइम हमारे पास इतना शॉर्ट था, अगर मैं किसी एक सॉफ्टवेयर को यूज़ करता तो देयर आर हंड्रेड्स ऑफ सॉफ्टवेयर्स, और क्योंकि वो अवेलेबल नहीं है फ्रीली, हम उनको यूटिलाइज नहीं कर सकते। अदरवाइज मैं किसी प्रोटीन को या वो जो सिमुलेशन वाला डेटा है हम उसको भी यूटिलाइज कर सकते हैं फ्यूचर में। बट फॉर अभी जस्ट जो अवेयरनेस के लिहाज से या उसकी जो अपॉर्चुनिटीज अवेलेबल थी उसके लिहाज से मेरा ये प्रेजेंटेशन का पर्पस था। बट आई रियली अप्रिशिएट योर जो इनपुट है, ऑफ कोर्स इट इज हेल्पफुल फॉर ऑल ऑफ अस कि हम इसको पॉजिटिवली यूज करें, ना कि इस टेक्नोलॉजी को ऐसे पर्पस के लिए यूज करें दैट इज नॉट मेड फॉर। थैंक यू सर। जी मैम।
I have just one comment, and I really want, there are very junior people are also sitting here, students, and we have to be really cautious when we use this ChatGPT. The other thing, as Dr. Saab mentioned, is that now journals has a requirement that have you used any artificial intelligence software for the preparation of manuscript. This is the disclosure that you have to give. The second thing is that when you submit a grant to NIH USA, they ask reviewer not to put any grant on the ChatGPT because it will be somebody's own idea which will be stored in their server, right, for some time, not for the longer, and it will make the job of the reviewer so easy that it will give a review report, and that would not helpful for the investigator and also not for the reviewer. So these are for the academia. I would say that we have to be really careful for making questions. If you make questions from ChatGPT for UGME or MPhil or for other, the quality of the question would be very questionable. So security compromise, and any student can use the ChatGPT and make the same question for himself. So आपकी quality of the education and everything will be affected. So you have to be really careful, and we should not encourage students at least to use ChatGPT for these purposes, finding questions and making. There are some ethical concern as well for the data protection for the patient. So for clinician, I'm sure they are aware. But in US, there's a lot of issues, and even in Aachen, we can't put any identifier or anything on any system or anything, any software. So these are my comments, and I.
Thank you so much, madam, for your useful insights. अच्छा, इसमें एक चीज़ मैं तमाम लोगों को भी जो यहाँ स्टूडेंट्स पे बैठे हुए हैं, don't write using ChatGPT for your manuscripts. उसमें सबसे जो इम्पोर्टेंट चीज़ है, it creates fake citations. इसमें जो भी साइटेशंस आएंगी वो फेक आएंगी। सो नॉट ओनली जो फेक साइटेशंस हैं, और बाकी जो चीज़ डेटा होगा वो भी जो है ऑथेंटिकेटेड सोर्स से नहीं होता। तो इस चीज़ को आप हमेशा माइंड में रखें।
Thank you. Can I just a comment and endorsing what all three of you have actually said. जब कैलकुलेटर्स आए थे तो I am Dr. Simon ji, I am sitting at the back, madam, if you can see me. अच्छा, okay. So जब कैलकुलेटर्स आए थे तो मैथमेटिक्स टीचर्स ने हड़ताल की थी सड़कों में निकल के अमेरिका के, कि मेंटल मैथ्स जो है वो बहुत ख़राब हो जाएगा और लोगों को बिल्कुल नहीं आएगा, इसलिए कैलकुलेटर इस्तेमाल नहीं करेंगे हम, और इनको मत लेके आएँ। जब हैंड कैलकुलेटर, ज़ाहिर कैलकुलेशन की बड़ी बड़ी मशीनें तो आ गई थी। टेक्नोलॉजी, यस, वी हैव टू बी कॉन्शियस। एक चीज़ जो आपकी बात से और इन दोनों की बात से जो है, टेक्नोलॉजी जब भी यूज़ करेंगे उसकी लिमिटेशंस अपने जहन में रख के यूज़ करेंगे। ऐसे नहीं हो सकता कि स्टूडेंट या कोई भी और कोई भी टूल यूज़ ना करे। अवेलेबल टूल्स यूज़ होंगे, लेकिन जो लिमिटेशंस उन टूल्स की हैं, जब जैसे जैसे वो हैं और जैसे जैसे वो बेहतर होती जा रही हैं, वो चीज़ें डेफिनेटली जहन में रखनी पड़ेंगी। और एनी सॉफ्टवेयर वो जो भी क्लेम कर रहा है उसको वैलिडेट तो डेफिनेटली कराना पड़ेगा फ्रॉम द एक्सपर्ट्स, या जो एक्सपर्ट है। देखें, मेयो हॉस्पिटल इस वक्त लार्जेस्ट, जो मेयो हॉस्पिटल नहीं कहना चाह रही थी, मेयो क्लिनिक्स जो हैं यूएस के, वो इस वक्त लार्जेस्ट बायोइन्फॉर्मेटिक्स रिसोर्स है, जो थाउजेंड्स एंड थाउजेंड्स ऑफ आपके एक्सरेज डाल के और अपने लैंग्वेज लर्निंग मॉडल्स को दे रहे हैं। सो आपके पास जो एल्गोरिदम्स किताबों में अब आ जाएंगे, वो किताबों में वो एल्गोरिदम्स होंगे कि रेडियोलॉजिस्ट को स्टैंडर्ड ट्रेनिंग देने की ज़रूरत ही नहीं पड़ेगी। और इसीलिए आप लोग सारे भी मेरे से बहुत पढ़े लिखे, और इम्यूनॉलजी मेरी उस तरह की फील्ड नहीं है, बट एआई के रेफरेंस में ये सारे मॉडल्स और सारी चीज़ें बनना शुरू हो जाएंगी। अगर आज का स्टूडेंट इन चीज़ों को नहीं देखेगा तो वो अगले 5 साल में पीछे रह जाएगा। सो टीचर्स से मेरी रिक्वेस्ट है कि यस, वैलिडेट कीजिए, यस, रेफरेंसिंग बिल्कुल दिखाए, देखिए। देयर कंसेंसस का सॉफ्टवेयर रिसर्च में बहुत अच्छा है। चैट जीपीटी तो उनको पहली चीज़ आ गई, सो एवरीबॉडी सेड कि शायद यही है। बट एनीवेज, देयर आर अ लॉट ऑफ एआई इस वक्त गूगल के सर्च इंजन में भी है, ऑल सर्च इंजन्स हैव इनकॉर्पोरेटेड एआई इन देम। सो जस्ट कि कौन सी चीज़ कैसी है, और उसने वैलिडेट एट द लेवल ऑफ द कॉन्फ्रेंसेस, जो नेटवर्किंग कॉन्फ्रेंसेस हैं, उसमें मेरी कंसल्टेंट से, सीनियर लोगों से ये दरख्वास्त है कि आप लोग प्लीज़ एक पॉलिसी गाइडलाइन ज़रूर निकालिए, जिसके अंदर इम्यूनॉलजी सोसाइटी की एक गाइडलाइन निकले, जिसमें ये लिखा जाए कि जी हम इस वक्त इस टेक्नोलॉजी को कहाँ देख रहे हैं और हम फ्यूचर में 5 साल बाद इसको कैसे देखते हैं। तो वो मेरी रिक्वेस्ट है टू द चेयर एंड द को चेयर कि उस पर ज़रूर कुछ देखिएगा। जज़ाकल्लाह।
थैंक यू सो मच मैडम, सो नाइस।
Just a brief comment. There are very well-defined regulations. So you have to use anonymized data, removing at least 18 identifiers, and then you use it. It is approved for use in radiology, a customized software, not everything. So that's what you have to demonstrate, that not the definitions of AI, but the actual use of AI, different softwares for a particular application. And then how would it gain the legal cover? And the preparation of manuscript question, that is a no. So never mention that. So that is just cutting your, I suppose, hands, feet and neck in the end. So never do that, and just go with the thought in the mind that there is a bill in National Assembly about personal digital data protection. So just keep that bill, read that, it is available. And whenever you are talking about AI, so keep the consideration of AI. They are specially strict for the use of data related to patients, much more than the personal data which has been stored elsewhere. So the patient-related data is much more sacred. So whenever you're talking about AI, prepare a talk in relation to its practical application, specific software, how it is used. So that would be clearing one pathway, and there would be others who would be clearing other pathways. So for the moment, the current scenario in Pakistan, have that bill with you, you can download it, read it, and if you're doing anything with EMR, the electronic medical records, then follow the guidelines, broad guidelines which are given there at the moment. That's not an act, but we wait for that. Till that time, be careful, be conservative.
Thank you, sir. Thank you so much.
Thank you, Dr. Fahim. After the lectures from the invited speakers, we are going to go on to the free papers. The time allowed for the free papers is 10 minutes each. think you, our moderator, would invite the free speakers.
Yes, ma'am. First is Dr. Uzair Abbas.
Assalamu alaikum. Thank you for having me here. I am Dr. Uzair Abbas, PhD scholar at Lahore University. The topic of my talk is latent tuberculosis infection observed to be lower in COVID-19 patients as compared to healthy controls in Pakistan. So since morning, a lot of immunology of COVID-19. So protection or severity from COVID-19 disease is largely a function of balanced innate and adaptive immune response. And early type 1 interferon response is very important for a protective outcome of the COVID-19 disease. And severe or fatal COVID-19 disease is a product of impaired innate and adaptive immune response. So the outcomes of SARS-CoV-2 infections and the protective immunity due to cross-reactivity from prior infections and other vaccination is also important to consider. And we all know that during COVID-19 pandemic, Pakistan observed a low CFR as compared to different neighboring countries, in which we observed 1.9% of case fatality rate of COVID-19 as compared to Mexico, Romania, Afghanistan, China, India. So we were at less for the CFR. So there are different factors to be considered for the protective immunity, in which there are co-infections like influenza, previous infection in which we have influenza, HIV, seasonal coronaviruses, and tuberculosis. Along with those, there is definite role of vaccination, which we have been discussing since morning, that is BCG and influenza. They also have a role on immune modulation. And then there are certain comorbidities like diabetes mellitus, hypertension, and cardiovascular disorders. So among all these, they have either protective or unfavorable outcomes with, when they interact with other different diseases. But we were more focused on tuberculosis, as Pakistan is high TB burden country among the in the world. So we ranked 5th among high TB burden, and the incidence rate is 250 to 300 per 100,000. Now there are two types of tuberculosis. That one is the active tuberculosis, and other one is latent tuberculosis infection. For those who counter the MTB pathogen, only 5 to 10% of those get active infection. Others either completely clear the infection or they leads to the latency of infection. There is a lot of data is available in the correlation of tuberculosis and the COVID-19 disease, but there is a little available for the latent tuberculosis, so we were more interested toward the latent tuberculosis. It is a subclinical mycobacterial infection defined on the basis of cellular immune responses to MTB antigens, but there is no active sign and symptom of the disease. So we can compare the X-rays radiograph of those. So this is the consolidations of the active tuberculosis. Then we have granuloma formation here in this, which is the hallmark of latent tuberculosis. So the 1/3 of world population is latently infected with the latent tuberculosis, and the latest population data is not available for Pakistan. There were few studies in 2020 and 2022 which have reported about 20 to 22% of the latent tuberculosis in healthcare workers, but general population data is not available. So if we talk about the immune response in latent tuberculosis, they have different immune profile as compared to active disease as well as healthy control. So mycobacterial tuberculosis, they certainly modulate the host immune responses, and it exhibits a Th1 cell response, which is also shared by the SARS-CoV-2 virus. And latent TB has different immune profile as compared to healthy control and active TB. They have different cytokine profile, they have different gene profile as compared to the healthy control as well as active TB. So we generated our objectives that how these individuals act when there is an exposure to SARS-CoV-2 virus. So the objectives were to determine the prevalence of latent TB in healthy population as well as in COVID-19 individuals, and to determine if there is any association of latent tuberculosis infection with the severity of COVID-19 disease. So for this, we performed an H.TB assay, which is an ELISpot assay, in which the T cell reactivity was known against MTB antigens in the form of spore forming units by the T cells when they released the interferon gamma. So a whole blood was extracted and PBMCs were isolated, and they were incubated for 20 hours with MTB antigens, and then the response were captured as these are the spot forming units in which The upper picture shows this positive sample of the latent tuberculosis, while the below is the negative. So these red dots are actually in the test well, which shows the interferon gamma releasing T cells. They are captured as a spot forming units, and more than 10 spot forming units were considered as positive. And we also validate this X.TB against quantiferon. So these are the results in which we found 17% of COVID-19 population was latent tuberculosis positive as compared to healthy controls. They were found 32% positive. So these are the tests which are performed by the Pearson's chi-square. And then when we divided these positive individuals in COVID-19 subgroups into, that is, one was the ambulatory group and the second was the hospitalized. So we found that all of the positive latent tuberculosis were found in ambulatory, and none of the hospitalized was found to be positive for the latent tuberculosis. So this is again the Pearson chi-square, in which we can see that all of the positive, 23 out of 128, were lying in the ambulatory group, and none of the hospitalized was found positive for the latent tuberculosis. So again, we then compared the interferon gamma response of MTB antigens in these individuals. So what we found from the t-test, we compared the healthy control and COVID-19 for the spot forming unit, that is interferon gamma response. So we found a significant difference between the interferon gamma response of these individuals, in which we can see this healthy control has high interferon gamma response against MTB antigens as compared to COVID-19 group. Then, when we divided these COVID-19 in ambulatory or severe to critical or the hospitalized COVID-19, so what we observed that there is high interferon gamma response in these individuals as compared to the hospitalized patient. So, moving toward the conclusion, there is high percentage of latent tuberculosis in healthy controls as compared to COVID-19 cases, and then interferon gamma response is seen higher in healthy controls as compared to COVID-19. And when we divided these COVID-19 subgroups in hospitalized or ambulatory, we found that there was a high interferon gamma response in ambulatory group as compared to the hospitalized. So we have seen an association that low T cell reactivity to MTB antigens is associated with severe COVID-19 disease. Then the way forward, on which we are still working, that there is a need of study to study the differential cytokine of these individuals in presence and absence of latent tuberculosis, and then there is need to study the differential regulation of immune pathways. As I have mentioned, that latent tuberculosis individuals has different immune profile, so there is need to see this association with the COVID-19, and then there is a need of study of differential regulatory molecules in which there are many genes, and yes, we are on it and we are working on this. In last, I would like to acknowledge my supervisory team, including Dr. Zahra Hassan, Dr. Kiran Iqbal, Dr. Rabia Hussain, and Dr. Martin Rottenberg, and then Tuleb Iqbal, who was very helpful during the experiments. Thank you. Any questions?
Thank you, Dr. Uzair. House is open for questions.
I have a question. So when you are doing this ELISpot assay or
Yes.
and the different gamma assay, you must
Yes.
have done the controls also, right?
Yes.
Negative and positive. So you've shown that the patients who had severe COVID infection, they had lower interferon gamma.
Yes.
So did you look at the positive control values also? Was there any difference? Because if they have, that means that they are not producing enough interferon gamma.
Yes, for the positive wells, we seen almost equal response in the positive wells. So these tests were done in a negative test and a positive. So for the positive wells, can I show you the picture over here? So the interferon gamma response. This was the PHA, yes, and so the interferon gamma response was same in all the positive wells.
Similar.
Yes, in 99% of the cases it was similar. For yes, there was some cases in which we had indeterminate results, so we have excluded those. But for these wells,
Those indeterminate results are important actually.
there were out of 275, only 4 we got indeterminate results. So there may be
मैं इसलिए कह रही हूँ क्योंकि देखो ये तो आप टीबी के खिलाफ देख रहे हो ना
Yes.
इंटरफेरन gamma. अगर वो आप ये देख रहे हो कि कोविड जो है उनको उसकी सीवियरिटी ज़्यादा है, in which it is the interferon gamma release is not okay. इसका मतलब ये है कि या तो आपको indeterminate मिलेगा कि आपका interferon gamma उसमें भी नहीं बन रहा होगा
yes,
with the PHA, और maybe the values जो हैं वो जैसे immunosuppressed patients के अंदर values they come down.
yes, So for the indeterminate, we observed the high interferon gamma response in negative levels also. So the indeterminates were classified as high interferon gamma response more than 20 spots in negative well as well. So we have classified the indeterminate on that basis. So we have got, yes, as per the manufacturer protocols.
मैं समझ गई, इसका मतलब ये है कि आपका जो कंट्रोल्स है उसके अंदर, जैसे मैं सोच रही हूँ कि वो इम्यूनोसप्रेस्ड है इसलिए उसके अंदर इंटरफेरन गामा नहीं बन रहा, इसलिए उनको सीवियर डिजीज है, लेकिन आपके पास आपने उल्टा देखा, आपने देखा कि
No.
हेल्दी की बात नहीं कर रहे, वो कह रहे सीवियर डिजीज के अंदर देखा ना.
No. I'm talking about indeterminates in all of the study cohort.
नाइस टॉक. आज मौका मिल गया सवाल करने का, बीबीएस में तो इजाजत नहीं थी. So I just wanted to know that you said that your cases had low interferon gamma, right? And they were critical. If they are critical patient, then even they have more lesser interferon gamma. So there comes a factor of the age. So could you tell that the critical were definitely they were older people, right? So in your conclusion, you have to put age as a you have to, for your modeling analysis, or I don't know, for statistical analysis, you have to put age as a factor because it could be a confounding factor for your low interferon gamma, right? And we know this that as we age, the T cell immunity goes down,
Yes.
right? So that could be one of the reasons. And for your future study, you have to adjust your result for the age as well.
Thank you very much. Nice work done. I just want to ask, आपने भी बताया कि जी 10 स्पॉट है जिसकी बेस के ऊपर आपने कहा पोस्ट है, तो ये वैल्यू आपने कैसे एस्टैब्लिश की?
These values were provided by the kit from the manufacturers. So they were basically, can I explain this? So we had two formulas provided by them. If a nil well has less than 10 spots, so the T minus N should be more than 10. So if the nil well has more than 10 but less than 20 spots, so T minus N should be equal to T minus N. But more than 20 spots in nil well, then it was named as indeterminate.
Thank you.
Assalamualaikum everyone. I'm Hafsa Faruqi and I'm first student of molecular pathology at Dhaka University of Health Sciences, and I will be describing a part of my study titled A Longitudinal Study of Immune Responses Induced by 3 different COVID vaccines in a Pakistani cohort. So this is a brief introduction about the topic. As we all know that SARS-CoV-2 first emerged in Wuhan, China in late 2019, and only a few months later WHO declared it as a pandemic when it spread globally. The etiological agent that we identified for COVID-19 was SARS-CoV-2, or severe acute respiratory syndrome coronavirus 2, and previously coronaviruses have caused 2 more epidemics, namely SARS in 2002 and MERS in 2012. And while there's been plenty of dollars of investments in finding a therapeutic agent for COVID-19, there's still no recommended standard treatment available. However, there are still drugs available that can manage the symptoms. History has also shown us that virus tends to mutate to better adapt to the hostile environment, in this case being the emergence of vaccines and the drugs that are being administered. But like most pandemics and most epidemics, a potential solution in wiping out the disease lies in attaining herd immunity among the population. And on to achieving that objective, several COVID vaccines were introduced in our population. However, these vaccines weren't tested. They were tested, but on a larger scale on other populations. So it is important to know how effective these vaccines are in our population using our demographics. So the objective of my study was to quantify and evaluate the concentration of the immune responses that are induced after these vaccines at different time intervals over a period of 1 year, and to study how demographics like age and gender affect these immunity. So this is just a brief overview that how traditionally a vaccine development process takes 10 to 20 years from when it goes into a phase 1 clinical trial to when it's available in the market. However, due to their urgent need, COVID vaccine development was very rapid and most of the steps were happening simultaneously, and the vaccine was available within 2 years of the pandemic. We also saw that there was an emergence of several variants, but more notably were the variants of concern, since they impacted vaccine efficacy and caused severe infection in most cases. So this also affects immunity levels. And the two major arms of immunity, the humoral immunity and the cell-mediated immunity, also affect our immune responses. Literature also shows that there are several host factors that influence immune responses, such as age, gender, nutrition, pre-existing immunity, and ethnicity, but here I'm just going to be talking about age and gender. Materials and methods of my study. We recruited 100 samples. Our sample size was 100, but we recruited 120. Since this was a longitudinal cohort study where we had to take follow-up samples, so to adjust for follow-up samples, we recruited 120. Those individuals were either vaccinated with Sinopharm, Sinovac, or Pfizer. We excluded those who got vaccinated with some other vaccine or were immunocompromised. The study duration was for 1 year, and we measured SARS-CoV-2 specific antibodies as well as interferon gamma concentration. The samples were collected at 6 different time points. First at baseline before the vaccine was administered, then 2 weeks after the first dose, 3 weeks after the second dose, and then 3, 6, and 9 months later. These are serum samples. This is the Roche kit that we use to measure this is a quantitative kit that measures the specific antibodies to spike protein for SARS-CoV-2, and we measured interferon gamma using the ELISA kit. Results. Okay, so here are the demographics of my study participants, where we see that there's a similar ratio between males and females. And with the study groups, most of our participants belong to the 18 to 30 age group, followed by 31 to 46 years, 47 to 62, and the lowest were the 63 to 78 age groups. Our data was not normally distributed, so I have reported here the median concentrations. And as you can see that on administration of the first dose of the vaccine, there was a gradual increase in antibodies. However, on the administration of the second dose, there was a sharp increase. And however, 6 months later, an attenuation in these antibodies was observed. And when we ran the statistics test, we saw that these differences in each time groups was statistically significant. However, for the interferon gamma concentration, the responses were more or less the same before the first dose, after the first dose, and after the second dose as well. And we also saw this data was not significant. We also saw the immune responses on the basis of each vaccine group, where we saw that all 3 vaccines are following the similar trend line, that there's a gradual increase in antibody titer till the 6-month time point, but there's an attenuation at the 9-month time point. However, out of all 3 groups, the Pfizer group had the highest antibody response even at the 9-month time point. On the basis of gender, we saw that males had a slightly higher immune response compared to females, and a man-white new test showed us that this was statistically significant. We also compared our results with other studies that have been reported, and for example, one in China, we saw that there was a similar response, but they too also saw there was a gradual increase after the second dose. However, 6 months later, the antibody titer started attenuating. Although we did not find any significant results in our interferon gamma, we saw that a study conducted in Bulgaria, they did. But this could be because they measured interferon gamma specific to SARS-CoV-2, while ours was a more general total interferon gamma concentration. A study conducted in Bahrain also had males who had higher immune responses, while a study conducted in Jordan showed that females had higher immune responses. And similarly in Sri Lanka, it was reported that those above the age of 50 had the lowest immune responses. Although we did not have any significant result in our age groups, we saw that those who were belonging to the 64-78 age group had the lowest titer. But we did not report this because we also had the lowest number of people in that age group. So to conclude, we saw that after the second dose of the vaccine, we observed the highest antibody titer, which signifies the importance of that booster dose. But even then, that starts waning after 6 months as well. We also saw that the older age groups are more at risk of attenuated immunity, so booster doses must be recommended, at least in that population, during when COVID is at its peak at least. And we also saw that COVID-19 remains viable if the transmission remains low. So I would like to acknowledge my molecular team, Dr. Saeed, Dr. Junaid, Dr. Miniba, the entire section at the University of Health Sciences, my friends and family. These are the references of my study, and I'm open to any questions that you may have.
Open for questions. Any questions?
Thank you very much. Very nice presentation. Well
Thank you.
Well prepared. Just one question. What is the interferon gamma, which is specific to COVID-2? Interferon gamma is just interferon gamma.
Yeah.
I thought it should be the same. The method of assessment may be different.
Yeah, the kits are different. They come, the antigen that they are responding to, they are specific. So they also follow the principle of ELISA, but some of these are specific to the parameter that you're measuring, the antigen or the pathogen that you're presenting. So they are more specific to that.
How can an interferon be specific for COVID-2? There is none. I think the method of stimulation may be different.
Okay. No, I was thinking something else. I thought the kit is different.
This will be last question for.
Yeah, very nice talk. I'm just wondering whether you have any correlation graph for the IgG response in the interferon gamma?
Yes,
How does it correlate?
We do. I haven't reported it over here. We saw that there is a correlation, but it was only significant among the first and the second time point, not among the rest. At the baseline and then after the second dose.
And there was Spearman
Spearman's correlation, yeah.
Excuse me. Thank you, Dr. Hafsa. Now I invite Dr. Abdul Qadir Parikh.
Assalamu alaikum everyone. I would like to thank the Pakistan Society of Immunology for giving me the opportunity to present this small work. The title of my talk is Correlation of Lymphocyte Subsets and Inflammatory Biomarkers with Disease Severity in COVID-19 patients. So, the COVID disease became a pandemic and it had a very adverse effect in overall health of the population. And the clinical manifestation, severity, outcome differed from one individual to another. There were around 15 to 20 persons which had severe COVID disease, where the majority of the patients had around 15 to 20% had severe COVID disease, while the others had minor COVID infections. And the increased rank you can see in severity has been reported with male gender, along with comorbidities, elderly age, diabetes, hypertension. So, it is also known that the coronavirus are known to have a profound effect on immune system dysregulation. When we talk about the SARS-CoV-1 and MERS viruses, it was known that they had a profound deregulation, and something similar was observed with the COVID-19 virus. So T cell and B cell alterations may foretell the severity of the disease, and a number of biomarkers were studied for the prediction of COVID-19 disease severity. So my study was to correlate the lymphocyte subset analysis and the serum inflammatory biomarkers with disease severity in coronavirus 2019 infections. It is a retrospective study, cross-sectional study, which was done in September 2021 to November 2021 in the Department of Immunology at Institute of Urology and Transplantation. The inclusion criteria was patients admitted to COVID-19 wards and intensive care units from June to July 2021. The COVID-19 infections were established by a positive PCR reaction by nasopharyngeal swabs. We excluded the patients, children, pregnant women, and patients with end-stage kidney disease and hemodialysis. Also, the kidney transplant recipients were excluded from the study. The patients were categorized into two categories: the severe group, which was named as Group A, which had fever, cough, along with radiological features of pneumonia, and the non-severe group, which had fever, cough, but no signs of pneumonia radiologically. We calculated the complete blood count with neutrophil to lymphocyte ratio, CRP, D-dimers, and serum ferritin. Serum cytokine levels for interleukin 2, 4, interleukin 6, interleukin 10, tumor necrosis factor, interferon gamma were analyzed by cytometric bead assay technique. Lymphocyte subset analysis or TB-NK panel was performed. And the data was analyzed using software SPSS 20. For the results, in the demography, as you can see, we had a total number of 54 patients. In the severe group, there were 29 patients, and in the non-severe group, there were 25 patients. The male to female gender was male were 33, female were 21. And in the severe group, as you can see, the number of males was 65% as compared to 34%. The median age for the severe group was 52, and the interquartile range was 43 to 65. Whereas in the non-severe group, the median age was 50 with interquartile range of 36.5 to 59 years. When you talk about the comorbidities, the severe group had 27% had diabetes as compared to 24% in the non-severe group, hypertension with 48%, and the non-severe group was 36%, COPD 6.9 and 8%. All these parameters between the two groups were statistically non-significant. The admission day was 5.3 days plus minus 1.5 days after the onset of symptoms. The presenting complaints between the two groups, cough was present in the severe group of 79% and 60% in group B, sore throat 25 and 15%. Diarrhea was 3.54% in the severe group, as in the non-severe group it was 16%. Myalgia was higher on the severe group, and fever was 100% in the severe group and 48% in the non-severe group. With the discrimination point between the two groups initially at the time of admission was multiple bilateral patchy opicities on CT imaging, which was 100% in the group A as compared to none in group B. Coming to the results, as you can see over here, with the leukocytes and the neutrophil count, there was no statistical difference found. But when we see the lymphocytes between the two groups, the severe group had less number of lymphocytes as compared to the non-severe group, which had 17.43. Similarly, this lead to neutrophil lymphocyte ratio derangement, which was high in the severe group as compared to the non-severe group, and it was steadily significant. The CRP was raised in the both groups, considerably more in the severe, similarly D-dimer and serum ferritin levels. When we talk about the cytokine levels, interestingly, we didn't find any difference between the severe and the non-severe group for all the parameters, interleukin 4, 6, 10, TNF-alpha, interferon gamma. Interleukin 6 was raised in the severe group, but as compared to the non-severe group, but it was not statistically significant. Some institutes have reported interleukin 6 being significant. When you talk about the lymphocyte subsets, as you can see over here, in 72% of the patient in the severe group, the CD3 cells were low, as compared to 40% in group B. The CD4 was low in around 80%, as compared to 48% in group B. And when we talk about CD8 cells, it was reduced in 55% of the patients as compared to 28% in the non-severe group. Similarly, CD19 was 27% reduced in severe group as compared to 16% in the non-severe group. When we talk about the absolute lymphocyte counts, you can see CD3, CD4, CD8, and B cells all were considerably reduced in the severe group as compared to the non-severe group, and these all were they were statistically significant between the two groups. There were mortalities. 7 patients died in the severe group, none in the group B. The mortality was higher in males, and the neutrophil lymphocyte ratio for these fatal patients was also sadly significant. So in summary, Mr. Jefferson, increase in inflammatory markers is seen in the severe COVID-19 disease, decrease in CD4, CD8 T cells and CD19 B cells count in severe group. Serum cytokine levels were not different between the two groups, and increased mortality in male as compared to the female in the severe group. In conclusion, pro-inflammatory biomarkers increase and the lymphocyte subset decrease in severe COVID-19 patients as compared to the non-severe group. In the end, I would like to acknowledge Dr. Khawar Abbas for his contribution for this work performed, and I would also like to thank Professor Dr. Rana Muzaffar for his ever support. Thank you, everyone.
Thank you, Doctor. Any question for Dr. Abdul Qadir?
Thank you very much. Nice presentation. It seems that if you don't have the facilities for lymphocyte subsets, you can just go with the absolute lymphocyte count and just relate it to the severity of disease.
Yes.
I just have this small query in mind that whether it happens in all viral infection or was it specific to COVID, and how did you differentiate between severe and non-severe? Was it just hospital admission or were there any other parameters?
So, sir, the severe and the non-severe group were differentiated and identified on the basis of patchy infiltrates in the lungs. Whoever had the clinically suspicion along with the CT imaging performed and with obesities, they were put in the severe group. All the others who didn't have the patchy obesities were put in the non-severe group. So that was the one thing. As compared to your another question, sir, regarding the other viral disorders, it has been observed that in viral infections, usually the lymphocyte counts goes up. But here in the severe group, and it has been reported by others also, that all the T cells and the B cells counts fall down in the severe group as compared to them. So it has also been reported by other groups also, and it's now in the standard. When we connect neutrophil lymphocyte ratio along with the lymphocytes, you can go with the severity of the COVID-19 infection. So these two parameters should be taken hand in hand with we interpret from this study.
Thank you. I think the ratio is just a mathematical reflection of increased lymphocytes. But what you are comparing is lymphocyte count in severe and non-severe.
Yes.
It is still higher than the sort of average expression of lymphocytes
Yes.
In the healthy person. So every hospital must have some idea that in this age group this would be kind of not the range, just the kind of 4,000 or 5,000 or maybe less than that, 3,000-4,000 is the absolute lymphocyte count, which is kind of healthy for, and then compare it to 7,000, which is non-severe. So it is still raised, but within range. It is increased, but not that much कि इसको नॉन सिवियर किया जाए. मतलब ये अपने हॉस्पिटल सेटिंग में कुछ अपनी लिमिट्स खुद ही डिफाइन करनी पड़ेंगी कि हो सकता है कोई और बीमारियां डिजीज बर्डन ऐसा हो कि लिम्फोसाइट्स काउंट्स उस एरिया में बढ़े हुए हो, तो न्यूरोलॉजिस्ट और सीबीसी एक नंबर ऑफ सीबीसीज को देख के एक थोड़ा सा एस्टैब्लिश कर लेंगे.
Right, it's a nice idea. और हमारे पास तो अल्हम्दुलिल्लाह मॉलिक्यूलर से इंटरेक्शन भी है, तो जो हमारे पास H1N1 वाले भी पॉजिटिव आते हैं really, but we do find something. So जो हमारे पास इन्फ्लुएंजा और जो भी पैनल है पूरा वायरल पैनल फॉर द रेस्पिरेटरी डिजीजेस, we can take that sample, identify from there with the positive PCRs, and as you said, उसको भी एक ग्रुप बनाए एक healthy control के साथ, and इसको compare किया जा सकता है. It could be done that.
We are short of time, so we can have more questions or any questions during the lunch break because we have to start the next session at 2 after the lunch break. Is that all right, Priyanka? I think we'll go ahead with the
Thank you, madam.
distribution. Okay.
Thank you, madam. So I will request So I request Professor Tahir Aziz to please kindly share shield distribution के लिए. जी जी सब मैच पहले शील्ड.
Probably you forgot because in the उसके अंदर आपने एंटी सेल्स के भी significant difference.
जी सर.
वो आप उसमें summary
Yes, that is a plus point. और सर viral में definitely है. Antibody dependent cell mediated cytotoxicity, NGS का role तो बिल्कुल है. नहीं next.
Secondly जो डॉक्टर ताहिर साहब कह रहे थे कि और वायरल इन्फेक्शन में भी एब्सोल्यूट कंप्लाउंड होता है कि नहीं. I don't know वो बाकी वायरल इन्फेक्शंस तो सर आप कैसे क्योंकि कोविड तो एक वेल डॉक्यूमेंटेड है, पेशेंट पकड़े जाते हैं. बाकी वायरल इन्फेक्शन तो चलती रहती हैं. I don't know.
फ्लू वगैरह देखते हैं ना जो मौसम आए तो उसमें करेक्ट किया.
So I would like to invite Professor Dr. Ghazala Jafri. Dr. Daud, sir, please kindly. Dr. Najia Iqbal. Dr.
Yes, sir. Okay.
I will request Dr. Najia or Dr. Surya, can anyone collect on behalf of Dr. Rabia, please, Professor Rabia Hussain.
पहले मुझसे खिंचवाने और फिर
Dr. Fahim Shahzad. Dr. Please kindly, Dr. Uzair. Dr. Hafsa Farooqi. Dr. Hafsa, I think she is in.
Sir, for Dr. Abdul Qadir.
Yes, sir. Thank you, sir.
Also for Dr. Abdul Qadir for moderating this.
Thank you, everyone. The session ends over here. Everyone is invited for the lunch, and after that, we will reassemble for the next session at 2 o'clock. Thank you, everyone.